I see it on YouTube.
Great, thank you. Excellent. All right, everyone, good morning, good afternoon, evening, wherever you are. Thanks for joining us this week for another fine seminar series. My name is Lauren Hayes. I'm a professor at the University of Tennessee at Chattanooga, and I'm pleased to be your host this week. Briefly, before I introduce the speaker, I want to just show you what is coming up. Next week we have Jacobus Boonsma, who will be speaking about eusociality. And then the following week we don't have the seminar series because of the holiday in the US and other countries. And then we have a couple more seminars coming up, Melanie Damhan, and then Manyari Jain from will be talking on the 10th of December. So I'll wrap up the fall fine seminar series. This week, I'm happy to host Nick Royle from the UK. Nick is an associate professor at the University of Exeter. He earned his PhD from the University of Durham in the UK and a bachelor's from the University of Edinburgh. He has an interesting research program that focuses on how organisms adapt to changes in their environment, both the social and the non-social environment. And he's particularly interested in plasticity of parental care and sexual selection. His primary study organism is the burying beetle, which I won't say much about because he's going to be talking about that today. But he uses lab and field work to address questions that relate to social living, parental care, and he has been moving more recently. Prior to the seminar we talked, he said he's moving more into more applied questions. So he's thinking about the influence of microbiomes on organisms, ecosystem services, and studying ecology and conservation of endangered ground beetles. Nick has an impressive record of publication scholarship. He has over 80 publications that span a wide range of really excellent journals. Some of those journals include, Tree, ProcB, all the good behavior journals, and he has at least one paper in Science. And he also has a book on the evolution of parental care that many of us are familiar with. It was one edited book from, I think it was 2012, Nick. I'm sorry if I forgot the year on that. But a really well-regarded book on parental care. And it's pretty clear that people value his work. You see citations, well over 6,000 citations of his work during his career. And so today I'm really happy to have Nick join us as part of the Fine. His title of his talk will be Social Plasticity and the Evolution of Parental Care in Burying Beetles. And with that, I'll turn it over to you, Nick, and thanks for being here with us today.
Thanks very much, Lauren. Can everyone hear me okay?
I can hear you perfectly fine.
Good. Okay, that's great. It's fantastic to be invited. I'm really honored to be here. I see got a few people I recognize either that I know kind of personally or kind of whose work I recognize, and that's really nice. Right, I'm going to start by hopefully sharing my screen effectively. Where is it? There it is. Okay, can everyone see that?
Looks good.
Yeah, that's looking good. Okay, fine. So you'll see that I've shortened the title slightly to get it to fit on, so I can have a nice big picture of burying beetle on there. Okay, so why are burying beetles so plastic? So I'll start by just kind of defining what I mean by behavioral plasticity and why I think it's important. So behavioral plasticity can be defined as the differences in behavior of individuals, often depending on the environment in which the behavior occurs. And these environments can be either social, i.e. kind of individuals interacting with one another, or non-social in terms of abiotic environmental factors. And individuals vary in their plastic responses to these environmental factors. And because variation is the raw material for adaptive evolution, if we want to understand trait evolution, then that requires an understanding of when and how variation in plasticity arises. And behaviors are highly responsive to changes in the social environment. And in the context of reproduction and parental care, these social interactions include things like competition for access to resources such as food or mates, female-male mating interactions, and interactions among family members, such as between parents and offspring or among offspring. And they're likely to be particularly kind of, important, these social environmental interactions, due to close links with fitness when it's in the context of reproduction and parental care. So behavioral plasticity varies quite a bit between and within species. So in terms of modes of care, there's a reasonable amount of between species variation. Some species exhibit all modes of care, such as Kentish plovers, which can have a biparental care, or male-only uniparental care, or female-only uniparental care. And similarly for fish such as Galilee and Peter fish. More frequently, there is, you might get kind of flexibility in modes of care, as occurs, for example, in red-winged blackbirds, whereby females always provide care and males sometimes provide care to some females. And so that's moving between kind of biparental care or female-only uniparental care. But in many species, there is no flexibility associated with modes of care. So blue tits, for example, and many other species of bird, it's biparental care is pretty much the norm. And there's as well as kind of variation in kind of modes of care between species, there is variation in the form and amount of care provided within species. So in a nice study of house sparrows by David Westneat and colleagues, they looked at kind of provisioning trips, provisioning behavior, and they found that it was repeatable across individuals. Overall, there was no sex differences, but there was individual variation in plasticity in relation to a couple of variables such as chick age and partner visit rate, but not brood size. And then this varied, so females were less responsive than males for some traits, such as brood size, but were more responsive for others, such as variation in nestling age. So what this shows is that there is quite a lot of variation in the form and amount of care provided within species. And some studies have looked at this in quite a bit of detail, looking at behavioral reaction norms. Burying beetles are potential kind of exemplars of behavioral plasticity. They've been providing parental care since the Cretaceous, some sort of 99 to 125 million years ago. There's fossil evidence of stridulatory files in which they used to communicate with their offspring on the abdomen, and which are typical of modern Necrophorine, the subfamily of beetles that Necrophorus vespuloides is from. And so they've been providing parental care for a long time, and there's quite a lot of variation between species in plasticity of modes of care. So the species that I study, Necrophorus vespuloides, shows all modes of care, whereas some other species, they only show one or two modes of care. And there is extensive within species variation in plasticity too. And the talk that I'm giving is kind of is predicated on the fact that this plasticity is related to three factors. One, that burying beetles are resource specialists. So they typically utilize the carcasses of small vertebrates, mammals and birds more often than not, as their source of food for offspring, and they use this as a larder in which they rear their brood, as illustrated by this picture here. Combined with that, they have there is very unpredictable availability of this resource in the environment. It's rare and ephemeral, and this is variation in what you might call a non-social environmental variable. And then there is a very dynamic social environment associated with access to that rare and unpredictable resource. So those three factors, I think, are potentially kind of very important in explaining why bearing beetles have such interesting and broad behavioral plasticity. So I'm just going to start. I mean, I know that Steve Trumbo's here, so he obviously knows a lot about a lot more than I do about burying beetles. But for those of you who aren't familiar with the life cycle, I'm just going to kind of walk you through it. So if we start up here, so typically adults have to kind of find a carcass and find a mate first, and so they're attracted to small decomposing carcasses, and then males, at least, if they find a carcass before there is a female present, will emit pheromones to attract a female. And once they've found the carcass, they prepare the carcass by removing all the fur and feathers and then smearing it with anal and oral secretions which which have antimicrobial activity. And then they will do this either alone or in a pair, and then they bury it and continue to kind of prepare it underground. And then they lay eggs nearby, which take a couple of days, 2 to 3 days to hatch. And then they crawl to the carcass. The adults have opened up made a little incision in the carcass there, and the larvae crawl to that, and the parents will then feed the larvae on the carcass. The larvae beg to be fed in a way that's analogous to sort of to birds begging as well, and the larvae can also self-feed on that carcass, and they spend maybe a week or so on that carcass being fed by parents, self-feeding until they are ready to disperse. And so at this stage, the larvae disperse. That takes about a kind of a week. They find somewhere to pupate, and then it takes another 3 weeks or so for them to come through pupation. And then when they close, it's another 10 to 14 days for them to mature. During this time, they typically feed as well, and then at that stage they are ready to go through the whole cycle again. So we can take advantage of this in the lab in particular, because it means it's relatively easy in order to keep them in the lab, and the whole cycle lasts about 7 or 8 weeks. So you can get through quite a lot of generations in the space of a year. So the species that I work on is, like I say, Necrophorus vesperloides, and they have biparental care typically, or uniparental care of either sex, so all modes of care. And the larvae beg to be fed using tactile cues. Obviously, this picture shows this happening. The larvae are reaching up to touch the mouth parts of the adult, which is regurgitating some food directly taken from the carcass. Obviously, this is all in good lighting conditions, but this would be happening. You have to remember, sort of underground, so it's all very tactile. And then begging peaks after about 24 to 48 hours, after hatching, then declines somewhat after that as larvae self-feed. And parents will directly feed the larvae. That's postnatal parental care. But there's also kind of prenatal parental care as well. This is that indirect care where they're preparing the carcass and keeping it free of microbes and defense of the carcass prior to burying it. But the work can be done not just in the lab, but in the wild. And quite a lot of the work that I'll be presenting here today has been done in the wild, mostly by this person, Paul Hopwood, who's a PhD student and postdoc with me. And he came up with this system, which he called microcosms, which is sort of a relatively kind of simple way using plastic tubing. You have a camera in the top, and then entrance and exit ports at the bottom that you can put mouse and or beetles, that have been derived from the lab into the wild, and then kind of do experiments using wild beetles. So that's a system that we had set up. So we've done this kind of combination of lab and field work in order to conduct our experiments. So this is a kind of cutaway showing the microcosms with a mouse on soil, and then the entrance and exit ports with a camera at the top we're filming. And just to give you an idea of the sort of footage that we can get from this, so this was in the wild. So this is showing interspecific competition. I mean, typically we imagine sort of interspecific competition being things like mammalian predators such as foxes, and or other invertebrates like flies laying eggs when they're attracted to their decaying carrion. But slugs are also kind of a major interspecific competitor, and I'll just show you a bit of the footage which shows how the burying beetles deal with these competitors. So hopefully you'll all be able to kind of see that happening. So you can see there's a pair here, and one of the pair is just about to attempt to remove this carrion slug from this mouse carcass that they are in the process of burying. See, the beetle has done a pretty effective job of removing the slug from the carcass. But obviously, when there are lots of slugs around, then they can often overtake carcasses. And we found that a lot of the carcasses that we put out were actually kind of totally overrun by slugs before burying beetles could utilize them. And these cameras can also kind of like allow us to witness behaviors that are known to occur, but rarely are seen, such as a brood thing. So this was an example of us, doing the filming in the lab. This was one of the early cameras that we had, so the quality is not great. But you can see here that down at the bottom, we've got the male, and the male is just processing the carcass, is actually kind of collecting some food for larvae, and the larvae are in this pit here, and this female is in the process. She was in the process of feeding larvae there in the pit, but she has pulled out one very small larvae, which she is now going to proceed to to cannibalize. And you can see it struggling away there, but you can see that how small it is relative to the other larvae in the brood ball. So she turns away in order to munch that larvae, which she will end up likely refeeding to its siblings. Meanwhile, the male comes round to feed the rest of the brood some regurgitated mouse from the base here. Okay, so what sort of sociological problems do burying beetles actually have to overcome then? So as I've suggested, they are resource specialists, and they specialize on this the carrion of small vertebrates, and this varies in its availability and also the size of the carrion that is available. And that varies both over time and spatially as well. As we've seen, there's a competition from lots of other species for access to this bonanza resource. And there is some evidence of carcass-sized niche segregation across species of burying beetle, for example. But some of the main competition obviously kind of comes from members of their own species. So intraspecific competition is also very strong. And that's because population density varies. So this graph shows dates of that beetles are active in the UK, and then the number of beetles that we captured in traps over time. And then we have kind of the solid line of males, and the dotted line are females. And you can see that population density varies quite considerably over the course of the season. So this is the initial adults that have overwintered, emerging and breeding, and then these are subsequent generations thereafter. And sex ratio varies as well. So there is a lot of variation in local competition, and that impacts patterns of parental care and mating behaviors. So one thing that is really evident for if you study Nicophorus vesperloides is there's a huge amount of variation in body size. So this is the pronotal width of beetles that are captured. So that's a measurement across there, the pronotum. And this is the dates of a population sampling, trapping, and each dot represents an individual. And you can see that you have some individuals that are twice the size in terms of pronotal width as others within the population. And the main reason for this is that this relates to carcass size. So this is the frequency distribution of adult size plotted on the y-axis, and then pronotum width on the x-axis. And these are the distributions of individuals reared on small carcasses of only 5 grams in size. And this is the distribution of individuals reared on much larger carcasses of 20 grams, which is more typical of the size that we use in the lab. And you can see that it's very closely related to carcass size. Larger carcasses beget larger beetles. And this matters because body size is really important in terms of direct competition for carcasses. So even relatively slight size differences between individuals of the same sex can impact contest success. So this is the probability of winning a contest, and this is relative male pronotum width. So here, where there is no difference between the individuals that are contesting, and they have a 50-50 chance of winning the contest. Whereas if you move just ever so slightly away from that, up to say like a 20% difference, then you have a near certain probability of winning any contest there. And these effects can be magnified when individuals are in relatively poor condition. So here, this is from a manipulation of the amount of food that they received following or during kind of sexual maturation, following eclosion. And individuals that didn't receive any food during that period are represented by this dotted line, and individuals that did receive food are represented by this solid line. And you can see that it is much more acute, the effect of the size difference between the focal beetle and the opponent when individuals are in relatively kind of poor condition. So body size matters when it comes to direct contests, and that's very important in the context of the ecology of burying beetles. So this is an example of what that looks like and how quickly these fights can be resolved. So you can see small individual here and a much larger individual. Larger individual immediately recognizes there's a small individual there, and they have a grapple. And I should point out that these, if that small individual doesn't get away very quickly, then it is potentially likely to lose limbs or antennae or even worse. So these fights are fairly intense, and the outcomes are often very clear if there's a big size difference between the two. So if you're small, then what options do you have? And one potential kind of option you have for success is to engage in alternative reproductive tactics. So both males and females can use alternative reproductive tactics related to their size. Females can mate with a resident male and then parasitize the brood. And they get away with this because if the timing is right, then the resident females will accept any foreign larvae because they can't recognize their own larvae. They just use simple cues related to when they've laid eggs. So this is actually kind of a male mating with the female. So you can see here, this is an intruder male that's come in, and this is the resident female, and he's busy mating with the female whilst the resident male is very close nearby, doesn't quite recognize what's going on at this stage. But then gets wind of the situation and breaks up the party, and then rather than chasing the male off, actually then spends time with the female and will mate with her accordingly. So alternative reproductive tactics are a potential way in which individuals can overcome the disadvantages associated with small size. And small beetles are less likely to signal, so emit pheromones to attract females, and they will tend to use these alternative reproductive tactics instead. So we looked at this in a paper with Maurizio Carter, and we used these selection lines that we generated for high and low mating rate. And mating rate is a paternity assurance behavior, and so we were using it to look at a whole suite of things, and this just shows that mating rates significantly diverge between those lines. And what we showed was that larger males signaled more than smaller males when there was a competitor, And highline males, so those ones selected for greater paternity or investment in greater paternity assurance behaviors, were more socially sensitive to size than individuals from low lines. And that's illustrated by this figure here. So this is on carcass signaling behavior. So doing what this male is doing here, doing a headstand and then emitting pheromones whilst in possession of the carcass. And you can see that this is the relative size of the focal male on the x-axis, and highline males, as you can see, were sort of much less likely to signal when they were smaller than the focal male they were against, but when they were larger, they were much more likely to signal than individuals from the low mating rate selection line. And this plasticity, this social plasticity, has a heritable basis. In other words, the selection regime times the social environment interaction was significant. So what does a signaling success look like? So here's another video which will kind of show this. And this was obviously in the wild. So you can see this male come in. You'll notice he's got some phoretic mites on his pronotum, and he's located the carcass that we've put out, and he is in the process there of doing that headstand behavior and emitting those pheromones. And you'll see that some of the time there's been a few minutes that have elapsed since he's been in. But after a relatively kind of short period of time, we get a female appearing, and the male then mates with the female. So from starting to signal, from finding the mouse to actually beginning to signal to a female arriving was only on this occasion about 3.5 minutes. Now that's kind of unusually quick, but it can often take relatively little time in order to attract a mate if there's one nearby. But obviously, kind of like when you emit pheromones, the other thing that you could attract are rival males as well. So there are costs associated with signaling too. So body size does matter, but is big always better? It turns out that when we did these experiments in the wild, and we put males out of different body sizes, it was such that smaller males were more effective at attracting a female first to the carcass when they were signaling compared to another male. And so this is a way in which they can avoid contest competition. So when they had sole possession of a carcass, they were better at attracting a female first. And so they were more likely to breed as a socially monogamous pair. And this is presumably related to pheromone emissions, and perhaps the composition changed slightly, or maybe females were kind of just more attracted to, there was something that signaled that they were relatively kind of small males. But regardless, it was smaller males that were more likely to attract a female first, and therefore they could avoid potentially fighting with these large males. Body size is also potentially kind of important when it comes to females too, not just in the context of signaling behavior. So there can be kind of individual specialization in resource use. So this figure shows the proportion of females that didn't breed on available carcass, and we put large females on either large carcasses or small carcasses, and also small females on large carcasses or small carcasses. And as you can see from the figure, large females deserted over half of the small carcasses they were given in the field at a much higher rate than any of the other treatments. And it turns out that large females do proportionally better on large carcasses than small females, whereas small females do much better on small carcasses. So this is the mass, total brood mass, and this is large carcasses, small carcasses here, and then small parent size. You can see that large parents do better on bigger carcasses, and small parents do better when there are small carcasses. So that's a fairly kind of rare example of environment matching, which is quite unusual. And this is probably because the beetles eat the carcass themselves, and so smaller beetles require less food, and so they tend to do a little bit better on smaller carcasses, and whereas larger beetles can lay bigger clutches and have larger eggs as well, so achieve a greater brood mass on these bigger carcasses. So in terms of what the kind of consequences are, in terms of plasticity, in terms of who finds the carcass first is a key determinant of the plasticity of mode of care. So if a female finds the carcass first, because she's usually already mated with males off carcass, she will be preloaded with sperm, and typically then she will just get on with burying and defending the carcass herself and processing it, and this will lead to sort of uniparental female care. And that happens in one of the few studies that's looked at this in around just over a third of cases. If a male finds the carcass first, if he attracts a female initially, if there is no contest at all, then you have biparental care. If the carcass size is very small, then some of the females may actually desert, and this is the likely scenario where you're more likely to get kind of male uniparental care, which happens in only a very kind of small number of cases. If, on the other hand, once a male has started signaling, he attracts another male first, then there will be biparental care, but with a contest, and that might kind of impact. So there could be opportunities there for alternative reproductive tactics on around the carcass, and then there might be associated costs to females with as a result of males doing this repeated mating as a paternity assurance measure. Okay, so that's plasticity of mode of care, but what about plasticity kind of among individuals related to kind of non-social factors? So plasticity is predicted to decrease with age because the benefits of responsiveness are likely to decline as individuals get older and the costs of reproduction increase. We looked at testing this in burying beetles, and we found that plastic response to a change in carcass size was less for older beetles, and this difference between young and old beetles was magnified when individuals breed first on a small carcass. In other words, there is some reproductive restraint. So these figures are a little bit kind of complex, so I'll just kind of walk through it here. So there is number of offspring on the y-axis, total mass of larvae, larval brood mass, and parental care provision on each of these panels. And then on the x-axis, we've got carcass size, large and small, and the arrows indicate the direction, so which one came first. So This was large, then small, and this is small, then large. And you can see that that overall, when you look at the impacts, that younger females tend to be kind of more responsive in their plasticity than older females for most of those traits. And age also affects plasticity of male reproduction too. So in this study with Kyle Benowitz and others, older males were found to have both higher mating and higher parental investment. So this shows male duration of care, and these are two treatments, one of which was a low paternity assurance treatment, the other was a high paternity assurance treatment, and then young males versus old. And you can see that duration of care was much higher for old males compared to young males, regardless of the paternity assurance treatment. And females mated to older males also reduced their own parental investment accordingly, because males were providing, a significantly improved amount of parental care. But overall, offspring performance was largely unaffected, because it is females that typically provide the bulk of the care, and they responded by dropping the amount of care that they provided. So as a kind of quick summary, there are sex differences in parental care plasticity. Females provide more care than males, regardless of whether they have a partner or not. That's illustrated by this figure here. So this is the amount of parental care provided under uniparental or biparental for females and males. They tend to be somewhat sensitive to male phenotype when providing parental care, for example, age. Males often provide no care at all. That certainly happens in a large proportion of breeding events. But when they do care, they typically provide more care when they're alone than when they're in a partnership. In biparental care, male behavior is largely independent of female behavior, but it is highly responsive to the behaviors of competing males. But offspring performance under biparental care is primarily determined by female care. So this is offspring performance. Duration of female care is much stronger effect than duration of male care in terms of the outcome. So given that, what is it that affects female parental care? So we expect social interactions in the context of mating might have important co-evolutionary consequences for the expression of parental care behaviors and vice versa. So how might sex differences in plasticity affect the evolution of parental care and mating systems? So we know from earlier that repeated mating is socially plastic and it can evolve. And conflict models, sexual conflict models, suggest that mating can be costly to females. So any behavior that increases parentage assurance for males could reduce the ability of females to provide care. So we tested this using our selection lines, this artificial selection on repeated mating rates that I introduced earlier on. And the results showed that, contrary to predictions from paternity assurance models, which indicated that males should increase parental care if their parentage assurance increased, selection for increased mating rate actually led to a correlated response in female, but not male care. And this is likely due to the potential kind of costs associated with superfluous matings for females. So you might get increased selection if there is increased selection for male mating rates as a kind of a social response to competition with other males. But this is likely to kind of like lead to a reduction in female care provision. And so there's likely to be kind of counter selection on male mating behavior if this imposes fitness costs on males. And so there's good evidence for a potential mechanism associated with that from Engel and Steiger in 2016, and they showed that there was an aphrodisiac, anti-aphrodisiac, sorry, called methylgeronate that's associated with a change in juvenile hormone production, and they both peak during provision of parental care, and this switches off the mating behavior of males and potentially makes it more likely that males will engage in parental care behavior. So that is a potential kind of physiological mechanism that could underlie this response. So what is male behavior sensitive to? Well, we've already kind of encountered that to a degree, in that it's typically kind of other males that they're most sensitive to. So we looked at this in a couple of papers, and we found that male care is highest when competition is also higher. So this is male care duration plotted against treatment. And this treatment is just pairs of males. This one has this treatment here has 1 male to 3 females. And this was equal sex ratios, but 4 individuals overall, and this treatment here was 3 males in relation to a single female. And you can see that male care is highest when competition is also highest, and the dominant male mating success, these are all the dominants and these are the subordinates, is positively related to the amount of time they care. That matches pretty closely with the figures here. And there was no benefit of offspring to offspring associated with the higher levels of care. And in fact, it was negative when there were lots of males in the treatment. So overall, this shows that male behavior is highly sensitive to that of rival males. And in actual fact, when they were providing care, so time spent actually kind of like, or duration of care, it was as much as anything else, it was paternity protection that they were engaged in. But it also meant that they weren't leaving to seek opportunities elsewhere. So why are berry beetles so plastic? I would suggest that it's not so much the costs and benefits of paternal care, as has been suggested by some others, that determine patterns of parental care here, but it's more the costs and benefits of maternal care, and allied to the costs and benefits of male mating behavior and the co-evolutionary relationship between the two. So males are generally more sensitive to the behavior of other males than their mates, whereas females are socially sensitive to both male mating and parental care behaviors. So does overall social plasticity maintain sort of co-evolutionary stability of parental care in the face of an unpredictable environment? So if I was summarizing, I would suggest that resource specialization and these co-evolutionary constraints, allied to kind of unpredictability in the size and availability of carcasses, favors social plasticity in burying beetles. And that could be kind of summarized as constraint is the mother of plastic invention. But what about microbes? I've not mentioned microbes yet. They're potentially kind of very important because they're the main competitors for access to carcasses that burying beetles have to deal with. And they both generate and contribute to plastic responses of burying beetles. So they're important at the various stages, because as the carcass is decaying, the microbes on it are reducing the nutritional value of the carrion to burying beetles. They're increasing the amount of toxins being released, but the volatiles that are produced aid detection of these carcasses. Then locating the carcass involves suppression of competitors and neutralization of the pathogens. So this is the burying beetle actually snipping off the hair of the mouse and smearing it with antimicrobial secretions. And it's this preservation. Here it is, kind of like in a nice brood ball, that with the aid of some friendly bacteria, that actually kind of is extremely kind of important component of the parental care of burying beetles. And then obviously consumption of the carcass following incision here, which is where the kind of the larvae are fed, and there is transmission of some bacterial commensal bacteria, such as Yarroia yeast, not microbes, not and bacteria, which can actually also act to compete with the pathogens on the carcass as well. So microbes are potentially kind of important in this process as well. They're the main competitors. And it's likely, and it has been suggested by Corner et al. Recently and others, that costly exposure to pathogens is likely to be a major selective force for social evolution. So burying beetles have to kind of act fast. They have to act fast to find carcasses and bury them to get them out of competition way. And they have ovoviviparity, so they're pre-prepared, they're young for rapid development. They prepare the carcass, smearing it with antimicrobial secretions to reduce competition from microbes. And then they feed their offspring, which increases growth and growth rate and reduces development time, so that they can get through this high quality resource as quickly as possible. So burying beetles use these antimicrobial defenses to suppress microbial competitors. Typically, the antimicrobial peptides and lysozymes they produce are like many natural kind of antimicrobial defenses. They're narrow spectrum, so they have a narrow mode of action. They're costly to produce. There's been work shown that increased production reduces fecundity and personal immunity. And they produce many different types of these antimicrobial peptides. Over 40 have been found for burying beetles, identified for burying beetles, but there are hundreds that are known in invertebrates generally. They have relationships with other microbes that help them compete with pathogens. And then they are regulated with regards to, they're upregulated when they're providing parental care. So they upregulate these costly defenses. There are sex differences, which relates to the differences in patterns of parental care between males and females. Depending on exactly kind of, what timing typically kind of males and females both take part in the preparation of the carcass, but males often do a little bit more, whereas females provide more postnatal care in Vesperloides. So one kind of key question that we're looking at trying to address, or have been relatively recently, is how have burying beetles been able to coexist so effectively with microbial competitors for so long, some sort of 100 million years, whereas humans have selected for increased resistance in target bacteria in less than 100 years. And in particular, whether it is production of a sort of diverse array of narrow spectrum antibiotics, antimicrobials, are used as a resistance management strategy to suppress these pathogens and preserve competition in conjunction with beneficial microbes that increase the fitness costs of resistance. And we've did a little bit of preliminary work which focused on 5 key antimicrobial peptides previously identified by other studies as being upregulated during the provision of parental care. And we looked at the variation in the expression of these AMP genes, and found that there was no sex differences. So these are all kind of different individuals along the bottom here. And there is males in bold, females in lighter coloration, and then breeders versus non-breeders in the different colors here. And we found that for breeders, defensin 1 was upregulated, which has previously been found. And there was co-expression of 2 of the others, so thaummatin 4 with C lysozyme 2 and C lysozyme 2 with defensin 1 as well, which suggesting, and this has been suggested that that there might be kind of synergistic effects associated with co-expression of antimicrobial peptides. And so there is clearly between individual variation is likely to be kind of both plasticity, a plastic response to variation in care, and also reflect potential underlying genetic differences as well. We found. So that's where I'm kind of attentively kind of taking this in collaboration with microbiologists. It's not my area of specialization, in order to kind of address whether plasticity and expression of these antimicrobial defenses and co-option of bacterial symbionts in competition for these carcasses, these rare stochastic resources, does that together promote the social evolution of family life in burying beetles? And with that, I'm going to kind of wrap up, stop talking, just acknowledge all the people that I've worked with, most of whom actually did all the research. So the people in black, people who have actually kind of done all the hard graft here, particularly kind of Maurizio Carter, Megan Head, Paul Hopwood have been kind of instrumental in doing all the work, and then other people are collaborators on that. So thank you very much for your attention.
Thank you, Nick. That was a really, really fascinating talk. I appreciate it. We have time. This is an opportunity now for people to ask questions. Just to remind you, if you got a question, please put a question mark in the chat, and I will take care of making sure that Nick has a chance to answer your question. If you're on YouTube, please post your question in complete, and either Adriana or Karsten will, or someone will let me know that there's a question in YouTube that needs to be relayed to Nick. And just a reminder, when you ask questions, please tell us your name and your affiliation, and maybe just a brief line about your research, just so we're trying to build community here. All right, so I'm going to start the question and answer session. So the first goes to Theo.
Yeah, hello, I'm Theo Bakker from the University of Bonn, working on fish, sexual selection and evolution. I have two questions. The first is about sexual selection. The pheromone release seems to be very effective for finding mating partners. Is there variation in the composition of the pheromones, and does it reflect male size or parental ability? Do you know that?
I don't know that. It's not something that I've looked at. It's presumed to reflect male size based on the results that I was showing you, but that is very much a presumption. I don't know whether Steve wants to kind of like jump in. Do you know whether there's any kind of good work on?
Yeah, I would push that off to Sandra Steiger, who's done more work with the pheromones. Yeah, I assume there are some differences.
Yeah, I'm not aware. I mean, I'm not aware of kind of any kind of evidence that it does vary with regard to size. Yeah, I mean, Sandra has done some work, but I'm not sure how much she's done.
Bigger males release more pheromones, or?
Again, it's not something we've quantified at all. And I mean, we would have loved to have done it, but I think we were doing this. The work that kind of like showed that there was an effect of male size on sort of being able to preferentially attract females. So small males being able to attract females was done in the field. And obviously kind of like pheromone work is very difficult to do in the field, if not impossible. And it's not clear whether if you brought them into the lab, you would even get the kind of the same response. So it'd be very difficult to translate that, I think, across between kind of like lab and field work.
I have another question on microbes. Why is there no resistance against microbes developing? Why is the other way around? Why do the microbes not develop resistance against the antimicrobe?
Well, I mean, I think that's a big question. I mean, like, we're suggesting that it might be for a number of reasons, one of which is related to the fact that they produce a diversity of these antimicrobial peptides. So there's lots of different antimicrobial peptides they could be producing. There's lots of individual variation. And so that's a kind of moving target, essentially. They're narrow spectrum, so they've got narrow kind of modes of activity. They're particularly kind of targeting particular kind of bacterial species, and there is also the potential for co-option of commensal bacteria, sort of friendly bacteria, which can also compete with these pathogenic bacteria. And so that's all likely to kind of reduce the ability of bacteria to kind of evolve resistance to the antimicrobial peptides. So that's the basis that we're kind of suggesting it kind of works, and that this is effectively a kind of a form of plasticity that allows this kind of moving target effectively. So, and it's something we're hoping to do. We're hoping to do kind of some experiments to actually kind of like look to see how increasing kind of competition from bacteria. So sort of, for example, carcasses that are much more mature tend to have sort of greater abundance of microbes. So sort of so abundance is potentially kind of important, but also diversity, which you can manipulate with antibiotics as well on the carcass to actually kind of reduce the number or the also diversity as well, and then look to see how the burying beetles respond in terms of production the antimicrobial peptides that they produce.
But the microbes are necessary for the young to feed on the carcass, probably, are they? Because they cannot, they need fluids resources, or how do they take up the carcass?
How do the larvae take up the carcass, you mean?
Yeah,
Or?
yeah.
Yeah, so the parents are sort of are actually regurgitating it, so they're eating the carcass. They're obviously preparing it, reducing the kind of microbial kind of competitors. And then the pit, the sort of like the little sort of incision that they produce there, they're lining it with their kind of antimicrobial secretions as well, and they also kind of they transmit these vertically to offspring. And I think I'm right in saying that that's now thought to be kind of more important mode of transmission than the sort of indirect effect of actually kind of like smearing it around the pit itself. But I mean, Steve, at any point, if because Steve's done much more kind of like on microbes than I have, so do feel free to chip in if I'm getting anything wrong here. I'm totally happy to be kind of corrected on any of this. But yeah, so that's how it works.
Thank you. Yeah, fascinating system.
All right, thank you. So I'm going to jump ahead to the first student question here. We try to encourage students to participate, and then I'll get back in the queue. So David, please go ahead with your question.
Thank you for a super interesting talk. My name is David Wood. I am working with Eduardo at Yale on energetics and behavior of biparental care in Tiki monkeys. So I am curious how you can distinguish males and females in all of these studies. And then I'm also interested, it seems like there is continuous variation in size in these beetles, but you've broken it down into the larger ones and the smaller ones. And I'm wondering if there's like assortative mating almost with the size based on the bigger females are more likely to abandon the smaller carrion bundles. And does that lead to bigger females mating with bigger males and vice versa, or not?
Okay, so the last bit of the question I can kind of certainly answer is that females seem to prefer small males in terms of kind of like on biparent, on the carcass. And I think that's mostly likely to be because they can physically dominate the males, so they're less likely to be harassed and suffer the consequences of repeated mating. So there's no evidence that I can tell that females actually there is assortative mating in that sense. Big males are likely to end up with females because they've contested and they've won out, so won access to females. But the evidence that we have is that females actually prefer small males, and I suspect it's because there is much less harassment, and it's also probably likely that a small male that has already got access to a carcass is going to be thanking his lucky stars that he has access to a carcass, and so he's perhaps more willing to provide parental care in that context, whereas big males likely to win out in any kind of a contest competition. And so, if they want to kind of shoot off, then, if there's reasonably high availability of alternative carcasses, then they would have a good probability of success on those carcasses. So, that's what I would say for the first bit. For the latter bit of the question, what was the first bit again? I've now lost.
How do you distinguish males and females?
So you can relatively easily do that like that by looking at the abdominal segment. So there's an extra one for males, and they're kind of much more kind of pointed backsides when you're looking at them. Females have a kind of round backside, and the sort of the hairs arranged on their abdomen is quite different. So it's relatively easy to sex them, and you can often sort of you could often sex them in the field, and with the cameras relatively kind of simply on that basis. I mean, there's a few that you get that are a little bit kind of more difficult to sex, so you have to have kind of close inspection. But generally speaking, it's really easy to identify the males from the females.
Thank you.
Great. Eduardo?
Thank you, Lauren. Thank you, Nick. Fantastic talk. And what a treat to have Steve as well. So we have the two experts. So fantastic talk. Thank you so much. So many questions. I need to go back and watch the recorded presentation to really get into all the details. I have a quick question following up on David's, and then one more substantial one for Dave. So what's the difference in body size between males and females? Is there, I mean, to what extent do they differ in body size or body
No,
mass?
not overall. No, there's no difference. Males and females are the same. They have the same kind of size distribution.
Okay. And then I'm going to, I think it was somewhere around the second third of your talk when you started talking about selecting lines for mating, the high mating and the low mating. And so again, that's why I'm saying that it's fascinating. You have so much information that I need to go back and go slowly through it. But I took some notes. So when you presented those slides, it's the first time that you mentioned selecting for lines and discussing heritability, you were talking about mating, you were talking about signaling, you were talking about body size or body mass. I thought it was interesting. Usually, my first reaction is, why don't we select? I mean, isn't body mass or body size kind of the candidate on which we may find the trait that, as you were saying, the trait that gets selected, and so we select for size to see what kind of evolutionarily relevant outcome the mating, the signaling, is favored by given size. But I just wanted to hear from you how you're thinking. I mean, we have a number of factors that are all interacting with each other, and in our minds, we're trying to put together this kind of evolutionary causation hypothesis, and I couldn't quite follow why you were not selecting for size to see how that size may be causing mating signaling, and also what's the heritability for size.
Size is heritable. I can't remember exactly kind of what the figure is, but most of the variation in size is actually related to kind of environmental factors. So kind of carcass size is the big one. Really. So that slightly kind of complicates things a little bit. I mean, when we were doing the selection experiments, there was no difference in the size of the beetles. So you would select on kind of repeated mating rate, and there was no difference in size between the kind of the low line beetles and the high line beetles or the controls. So there was no kind of like correlated effect there associated with repeat mating rate, and that kind of doesn't surprise me given the kind of the ecology of burying beetles and this flexibility in that something like repeated mating rate is going to be important regardless. You're either kind of you would either in the context of alternative reproductive tactics or which is typically kind of like when you're kind of smaller than your competitors, or if you're actually kind of if there's contest behaviors and you've had to sort of fight for access to a female that might have already mated with other males, and so this repeated kind of mating is going to be important regardless of your size. So I think size in that sense is probably kind of a bit disassociated from repeated mating rate.
Thank you. So much information to digest. It's fantastic system. Fantastic system. Thank you so much.
They are nice.
Great. So next we have Clara.
Thank you. Clara Jones, Clara B. Jones, Social Evolution, retired. Thank you for the talk. Very interesting in my opinion. I have one point of clarification and then a brief question.
Mhm.
My point of clarification is you stated that offspring did not benefit from either of the two conditions, parental care, no parental care. Does that mean that you measured mortality in offspring?
Was that, sorry, what, was
Does
that in
that
relation
mean
to, I think that might have been in relation to, I'm trying to look through.
you found that there was no difference in um, survival of offspring in either condition? Is that not correct? In other
Um,
words, offspring didn't benefit from either parental care or non-parental care.
we didn't have, I didn't present anything that was kind of no parental care versus parental care. I mean, like, so this is a species of burying beetle that that does require parental care. I mean, it's so I mean, like some work, Rebecca Kilner and others have done work actually looking at kind of no care selection lines versus kind of, you know, care, but it's not entirely kind of like no care in that, you know, they do kind of open up the carcass and kind of like make it available, and so there's a little bit of kind of a preparation in that, but my none of my work was looking at kind of no care versus care. So
Okay, I'm not being clear. Let me move on. Do you find any general principles from your work on a single species?
well, that is a good question. I would say that the kind of the general principles were kind of outlined in terms of like the importance of, it's really the sort of importance of maternal care rather than paternal care, and the influences on maternal care as opposed to the relationship between male mating behavior and male provision of care. So I think that's what I would say was the kind of like something that is generalizable away from kind of burying beetles, but yeah, that could be debatable, I guess.
Okay, thank you.
Okay, next we have Zulema.
Yes, hi. Sulema Tang Martinez retired from the University of Missouri in St. Louis. And while I was still active, my work was primarily on social behavior, communication, individual and kin recognition, primarily in rodents, and also some conceptual work on sexual selection. And I have a whole bunch of questions. And Lauren, if I go too long, stop me, because I don't know how many people are after me.
It's just me, so go ahead, Zulema,
Okay.
please.
Okay. So my first question is, do females ever compete with other females, like especially on large carcasses? Do you ever have more than one female being attracted? And then do the females compete and try to keep other females off the carcass? And
Yes.
you know, I can move on to the other ones because they're not necessarily closely related. So that was the first question about female competition.
Yes, there is female competition, and so some of the work we were doing on the effect of nutrition on contest behaviors was on both sexes, and you got exactly the same kind of like effects in males as in females. So typically, females will fight with only with females, not exclusively, but nearly kind of exclusively. I mean, the pairs can sometimes kind of like potentially kind of gang up, but males are typically kind of quite keen on, you know, not fighting with any interloping females. So yeah, there is kind of female kind of contest behavior, and that's because as I mentioned kind of briefly, any kind of interloping female has the opportunity of, if she's there in time, in good time, of potentially kind of like laying her own eggs in the soil nearby. And if she gets that timing right, and it's around about the same sort of like time that the resident female has laid eggs, then it's likely that you know, her offspring will be reared kind of, you know, sort of by the residents, so kind of your classic brood parasites. So yeah, you do get kind of like you know, very much kind of competition, you know, among females in exactly the same way as among males.
Thanks. Then my second question is there any evidence that females that mate with multiple males have higher reproductive success? And also, I guess related to that, is there any evidence of order effects in mating for the males? Like, is there, you know, first male advantage or last male advantage or not?
Again, because I haven't kind of worked specifically on kind of on multiple mating per se. I mean, like it's a major feature of kind of burying beetle ecology, in that you know, females that are caught from the wild are nearly always mated with a male beforehand, or you know, if they arrive at a carcass, and we don't necessarily know kind of like where they have done, but there is evidence that they will mate with males opportunistically, which makes good sense, because if they have kind of sperm, if they're preloaded, then if they find a carcass, they can just get breeding immediately. They don't have to you know, hang about until a male comes, and then, you know, suffer the potential costs of contest competition there. I don't know myself whether there is evidence that there is higher success in terms of breeding success if a female is mated with more than one male compared to a single male. I haven't kind of done it myself, and I'm not sure. I mean, it probably will have been done in burying beetles, but I can't think off the top of my head whether that's the case or not. I'm afraid.
Yeah, and I suppose that also means that you would not know about male mating order effects.
No, I think, but again, I would have to kind of like double check this, that it's last male precedence, which makes sense from the point of view of kind of like their repeated mating behavior. You know, if because they really are, you know, if there's been, so you saw on that kind of like a bit of video footage when there was you know, another male kind of interloping, then the male was immediately kind of like on the female and repeated mated for a long time, and you could see whenever there's any kind of male kind of interactions, then repeated mating really kind of like flies up. So I would expect that there probably will be kind of last male precedence.
Okay, and then the third general question is, you talked about they're preparing the carcass with antimicrobial agents, and about they're removing slugs and so on. But I noticed on several of your pictures that there also were flies on the carcass. Do they remove fly egg or any other insect? But I'm thinking of flies, larvae or eggs. And in one of your videos, I noticed little insects running around, tiny little insects compared to the bearing beetles. Do they also try to keep the carcass free of eggs and larvae from other insects?
So the main defense they have against that, and again, Steve, kind of, you know, chip in if you want to, is in burying, kind of like burying things as quickly as possible, getting it underground. So burying beetles typically they decaying carcasses are kind of are easier to find, but they're obviously less attractive. They're potentially kind of costly to use, and so they prefer, if you give them a choice, kind of typically kind of fresher carcasses for breeding on. They like kind of older carcasses to eat, but they for breeding on they prefer sort of fresher carcasses. So there's a kind of sweet spot from that point of view. And flies are typically associated with further stages of decay. And so if there are already kind of like lots of flies on there and fly maggots, then that probably means the carcass is spoiled as far as the you know, berry beetles are concerned there. Steve, do you want to chip in? I know you just...
Yeah, I mean, if there are a few fly larvae, then the beetle might just kill them a few. But if it can get overrun, then they just abandon it, as you say. Also, the mites on the beetles will sometimes come off and will kill the fly eggs. Again, if there are a few of them, if there are too many, that's just too difficult to control. Some of those things you might have been seeing in the films crawling around the carcass might have been the mites as well in the videos.
Yeah. So do the mites jump back on after they're finished killing or doing whatever they're doing on the carcass? They jump back on the bearing beetles then?
Yeah, I mean, yeah, they're phoretic mites, so they use them as like a bus system, sort of to get around.
Okay.
But I mean, I'm sure you will have seen kind of many more than me. But in the wild, where we are, there's sometimes you find burying beetles with so many phoretic mites on them that they can't fly, and they can't actually sort of. I mean, there's just huge numbers, and you get in the lab if you don't remove kind of all the phoretic mites that you know, like if you just leave one on, you know, like a single individual, then you know they can, if it's a female, that you know they can proliferate really quickly on a carcass, and you can get large numbers. And what I find kind of most fascinating, and I'm not quite sure how they know, but there's definitely they very good at monitoring the behavior of burying beetles, in that they can be, you can see them, they're on the carcass, and then the burying beetle is clearly decided to go. It's about to kind of, you know, leave the carcass, and as it is going, the mites just sprint over and like jump on. I mean, like, it is phenomenal to see. They can be swarming all over the carcass, and then they just kind of like shoot over and jump on the burying beetle before it shoots off.
Yeah, fascinating. And then my last question is really simple. You talked about old and young beetles. How long do they live? Do they live multiple breeding seasons, or?
No, so an old Vespuloides anyway is about kind of 3 months old.
Oh,
that
wow.
would be quite old. I mean, you can get them living a little bit sort of longer, but typically it's yeah, 3 months would be fairly old, and you know you do get sort of mortality kind of like increasing from about 2.5 months or so.
Wow, that's really short.
Yeah, which is kind of crazy, because I mean, like, you know,
Yeah.
in some other respects they have kind of like slow life history characteristics, as in they have this extended complex parental care and things, but they live their life at sort of, you know, a bit of a race. Although, I mean, that you do get the ones at the end of the season diapause as adults and then re-emerge and then breed. So obviously kind of that'll be 7 months, 8 months, you know, that they're alive, but obviously they're, you know, in a state of quiescence for a bit.
Well, fascinating. Yeah, it was a great talk. I had the other question I had, except that you already answered it, was the one that Eduardo asked about heritability and of size. And I was thinking, wait, you know, we have heritability of size, but it seems to be dependent on the size of the carcass. And so you answered that question already. But it was great talk. Really enjoyed it. Thank you so much.
Okay, thank you.
I don't see any more questions in the queue. Feel free to pop more in there if you have them. In the meantime, I'll ask one, Nick. So you showed infanticide that occurs in these animals, and I'm curious, what are some of the explanation or causes of the variation or correlates to the variation that you see in the extent of infanticide? So, for example, is infanticide associated with paternity assurance? first of all, do males engage in it? And if they do, is it associated with paternity? And then also, is there differences based on sort of like brood size and things like that?
I'm not sure that males are known to engage in it. I think or certainly the only direct evidence I have is of females kind of, and it relates to kind of brood size. So adjusting, I mean, typically in kind of like in many birds, for example, with the hatching asynchronous, you get overproduction and then kind of subsequent kind of readjustments. Where we've got kind of like staggered age and depending on kind of resource availability in the wider environment, you can either sort of raise big brood or kind of like get rid of them relatively cheaply. And I think effectively that's what burying beetles do. There's always a little bit of kind of overproduction, perhaps associated. I think somebody was suggesting that there's sort of they lay a reasonable number of infertile eggs, as a sort of as a bit of a hedge against kind of reduced success there. But if they do really well, and they probably laid more eggs than they can raise in terms of larvae, because when you see, I mean, like I don't know whether you've seen this, Steve, but kind of certainly kind of in the wild, you can see them. When they first find a carcass, literally. I mean, I've been there when they've you put a carcass down and they first come around. They quite often kind of like go around it. They sort of like they sort of go. It's almost like they're kind of judging what size it is. So they're actually kind of like checking out kind of like how big the resource is itself. So they seem to be pretty good at kind of judging that, but obviously there's quite a bit of individual variation in the number of larvae they have and the number of eggs they lay. So I think most of it is related to that kind of like brood adjustment, and the removal of larvae that are obviously not going to do well. I mean, in that video, that was a kind of really clear example. Most of the brood were really quite chunky at that stage, and then there was one really tiny larvae that clearly wasn't thriving, and so removal of that individual was I think the sort of a typical form of infanticide.
Yeah, just to say a couple things. When they first arrive, you're like they crawl around. It's almost like a preliminary sort of assessment, and then it's almost like they get more cues about well, first of all is like, are they going to accept the carcass or not? If it's a huge carcass, they'll abandon it, but they may make sort of a later adjustment by continuing to, after they've rounded the ball, to crawl around it. Males, if you take away the female, the males will adjust the brood. They will do that sort of infanticide. I don't know. Actually, one thing that we're looking at in our lab now is thinking about what happens when you have the male and female. Do they both get involved, or suppose they have different ideas about how many larvae to raise? We're sort of, that's one thing we're actually sort of thinking about doing in our lab now.
Very interesting. Thank you. I think there you are. Got moved around on my screen here, so I got disoriented a second. I think Ruchira, you have a question? Please go ahead and ask it.
Yeah, hi. I'm Dr. Ruchira Sen, and I work on social wasps. And in 2006, I mean, it was a fantastic talk, and I really enjoyed it. And in 2006, I had done some work where I had shown that male wasps, like I work on social wasps, so male wasps actually feed larvae if the females are removed from the nest. So, and that work was kind of very different from what is known in social insects. Now, today I heard this talk where I see biparental care again. So I'm really fascinated. I'm just interested to know, in other insects, do you get to see biparental care? Like, in how many insects do we see? I mean, isn't it really, really rare?
Yes, it is fairly unusual. I think most care in sex is female only. I mean, like, so certainly things like earwigs, is female only care. and other species. So yeah, so it is very unusual sort of biparental care, and I think that relates to the kind of bonanza status of the carcass. You know, it's such a big kind of resource that there are definite kind of like benefits associated with kind of, you know, the male getting involved, and the fact that the probability of him finding another carcass and being able to kind of breed elsewhere is really slim. I mean, they're funny because like you know, if you walk in the woods, it's not exactly kind of like littered with dead bodies as far as you can make out. But you know, the population of burying beetles that you can capture indicates that there must be reasonable numbers. But there's always a lot of competition. You know, if you put a mouse out, you know, then there is always a lot of kind of competition for access to that resource. So they're a bit weird because they respond in the lab. You know, you can breed them multiple times, but and the life cycle is enough so that you can sort of breed them 3 times. You know, typically, but in the wild they must be functionally semiparous. You know, so I mean,
mostly it is single parents. I mean, this is one unique case that I heard in insects where, I mean, I don't know how unique it is, but as far as I have known about insects, it's mostly that it is always uniparental and mostly the mother showing the care, right? So it's a fantastic system, and I really like the talk. Thank you so much.
Well, thank you.
Can I say something?
Yeah, please.
Jim Hunt, many years ago, I believe, also observed at least a few cases of male social wasps feeding young. And I can't remember the conditions. I don't think that he had manipulated the colonies to remove females, but he did see, I'm pretty sure it was Jim Hunt, because he was a colleague of mine in the same department. And I believe he did see very few, but some cases of male wasps feeding larvae.
At the same time as females, or when the female was not?
Yeah, the females were
there.
Because like I said, he didn't remove females from the hive. I think he was just sort of stunned to suddenly see males engaging, any males, you know, and again, I think it was just a very few cases, but he did see some males that were engaging in care of the larvae. I'm pretty sure it was feeding the larvae specifically.
Yeah, actually, Jim Hunt did do some work like that, and many other people have also shown that in polysters, males do feed larvae once in a while. But what we did was a more systematic experiment where we had given extra food to the males and we had removed the females from the nest. And then we showed that in absence of the females, the males are getting an opportunity to show the parental care, and they do it. And another similarity with this work was that under the care of the males, the larvae did not do very well. So under the females' care, the larvae actually survive well. And the females feed all sorts of larvae, but males only feed the biggest larvae. And they don't do a very good job, but they do it. In social insects, it's like rare occurrences when the males put some food in the larvae's mouth. And we had shown that no, it's just that they don't get the opportunity to do it. So if you remove the females and give extra food because they don't forage, then they can show some extra care.
Great.
I mean, with the burying beetles, I mean, like they do have division of labor, you know, so that's what I think is kind of particularly interesting, is that in a biparental situation, then you have kind of females doing kind of most of the postnatal care, males doing a bit more of the kind of prenatal care. But if you then kind of remove one of them, you know, those individuals, then the other individual can do all jobs. So they have that flexibility, you know, and they males actually kind of can do pretty much as good a job as females. It's just they don't. So they have the capacity to do it. They have the facility. They can do it if they're kind of forced to. And occasionally, like in the wild, you get females deserting and males being kind of like left to carry the can. And when that happens, they're perfectly able to do that. And presumably in those situations, it's typically kind of like once females have already kind of like been involved in kind of processing the carcass and stuff, and so the actual kind of like costs of kind of finishing off the job are probably relatively slight, given compared to the potential for further opportunities elsewhere.
Great. I think what I'm going to do, Nick, is I'm going to end the YouTube stream. So I want to thank you for a wonderful talk and for a great discussion. Appreciate you being with us here today.